We didn't use the wallet to understand. It's everyone to it. I talk about
So what did you do instead?
we got to play.
Joking.
What did you do? Maybe you did.
Oh, no, it is working. I was sick.
Just watch out for the, we're gonna live stream it. So,
Oh,
yeah, but if you want to get in, maybe just take off the charger.
Yeah.
The live stream is that gonna be? No, that's gonna be back.
Yes.
The whole time.
Yeah,
Yeah, we're both here.
Hello.
Never mind.
Hey. Oh, yeah. Sick.
We ended up. I wonder if it's really fun. What'd you say? No, it's, I think it's good. Do you want to go down further? What's this? No, I think it's fun. No? Yeah, I think it's fun. I don't know. I was also thinking about London. It's very steep. Yeah, I know. Just things that be like bad, like narrow and really narrow. Yeah.
Yeah, especially. They're good. And I was always joking about the name. After showed it. And just to see like this. About a sport, Ninja Tech sport company. They have the same icon or logo.
Right, what did it say?
Did you realize?
Yeah. What? What did it say there? Yeah, that's why I didn't want to start.
Yeah, because
Yeah. Like,
but you could sit on either side.
but then we were like, yeah.
Like if you just sit
everything's the
directly
same.
here, then you might be in the field of vision, but you could sit either side.
Yeah. We're not using the microphones of the computer, they're using.
No, this is just for recording this.
Yeah, but I mean, what we say, is that gonna be.
Possibly, yeah.
Or do you think they're using microphones?
I don't know. But
Where did everything go?
I think to the toilet.
I wonder if you want to just understand that I'm gonna do it. Oh, something like to, yeah. This is fine. Do you guys want to be here? Should I?
Yeah, I might just come.
Okay, cool.
Using that laptop
You want to go over there? Yeah, let's go. We can sure. Yeah.
Wait.
You can sit here. It's very down.
But look.
So let's go inside.
Yeah, Yeah, I thought about my phone died. Handwriting.
Yeah, I think so. Oh, absolutely. That's what you
You want to say
Yeah. Go in front.
Is this picking up the sound or is it just the mic?
That's just for the
live stream.
live stream. Yeah. Hey guys,
So
do
we
you mind
said
if you guys sit like further forward because we have like live stream here and like a shakes and stuff.
Oh, yeah. Oh, we can sit here?
There, or it's better in front. You know? I mean, look at all that space. So much.
What do you expect? You guys see here? Is that pasta? All gross? Yeah. Alles pasta. Yeah. Come here. Yeah.
That's possible we have one.
They didn't want you to sit in because we're live streaming here, so they wanted stream. Yeah. So, yeah.
Is it available here? Is it?
Yeah, that's fine. Just like right in front of Yeah, exactly. Oh.
Are you by your camera just in case?
Just I guess both. Just maybe we put this beside you.
I think they were saying there because everyone's shakes.
There. Yeah, I think so. You guys just keep an eye on this. Right, yeah. So yeah, good. You sit there and you can be the person that's making sure no one. There, then. No, well, just because this is. Well, you can sit there.
Oh, so you want to sit
Okay.
near that seat.
Okay, go. Just be careful of the laptop.
也不好走啊。
Okay, sit inside.
Sit there.
I think do you want to get, I don't sure if you want to sit in.
Yes, yeah.
Yeah. I need to leave.
Ah, okay, cool. Thank you.
是呀，是呀。我可以进来，我要进来。进。
Oh, sorry.
You are too tall.
Yeah.
Yeah. Sorry guys, could you not sit on this table because we're live streaming this? So you're shaking the table.
Sorry?
Could you like sit in the front because you want to shake the live stream using a laptop and streaming?
Oh, okay. I will shake the table.
Yeah.
Oh.
What's that?
It's Shake's Table. We're doing a live stream to other places. If it's cool.
What, you mean the whole thing or just this one?
Actually, it should be okay for the other one. As long as it's not on this one. Sweet.
You wanna go?
Yeah.
Cool. Yeah. I think so. We're being live streamed now. I think we're doing recording as well. I'm not actually sure if we're doing recording, but it's being streamed. Try and find out. You want to write down your email? Then I'll try and find it. So I'm not sure if it's being recorded. I think it's just being live streamed. Yeah, it's just streaming.
Okay. Back here, I'll take it. Same.
Oh, you know someone sitting here? Okay. Would it be possible to sit somewhere else that might block the screen? Like, thanks.
Oh, there's a camera. Yeah.
Broadcast, right?
1, 2, 3, 4.
Sorry, do you mind not sitting here? Just get the live stream is here and the table shakes.
Oh, right, okay.
So there should be space in the front.
All right.
You guys packed.
Yes, stop it, just fine.
Put it here so that you don't have to stand there. Good shot. Yo, not here because of the table. It's been used for live stream, so it's like shaky. So unless you're super careful and don't touch the table.
Running by.
What?
Come on.
You can jump.
Yes.
How do you hold, like, how do you do in your high school?
I don't know.
See the down there.
Maybe they're downstairs.
This mine? Oh, yeah. Oh, it's Patricia.
Hey, I'm sorry. You Okay, yeah.
Chat. You apps.
Yeah.
If you sit here, could you be like careful? Yeah. Get down in front would be ideal.
Yeah.
Myself. But I don't trust anyone, man. Oh, you can sit here if you want, but just don't shake the table.
My handshake. I said,
Oh, I'm sorry.
I'm not gonna say.
It's only these six. All of them. All of them. Oh, okay. Is there anyone else in there? No?
You want you really serious if you want I can
Gut. Oh ja? Yeah. Yeah.
Oh, but that's just like right next to it.
What will be great?
Hi.
Huh?
Ich arbeite in Cambridge. Nee, ich bin ja kein Student mehr. Ich bin fertig mit MIT.
No, we want to. No, we need to leave.
Ja, me too. Das ist verknüpft mit solchen Workshops. Aber ich bin jetzt Microsoft Research Treffen halt hier. Voll, also das Headquarter war fantastisch und jetzt habe ich halt nach Cambridge gewechselt und jetzt bin ich seit 2 Wochen offiziell. Das kann man noch schwer sagen. Ich habe ein eigenes Projekt, das ich gerade so ein bisschen anschlagen, was noch immer halt mit Biodesign zusammenhängt und dann bin ich noch in der Grand Challenge wahrscheinlich drin, was für Explorer-Ding ist. Nee, ist gut. Wie geht's dir? Bist du jetzt wieder da?
I want to sit close to the exit.
Lass später mal quatschen.
Okay. Yeah, we're all waiting.
Yes.
I'm not gonna say
Well, hello everyone. Can you hear me? Hello? Oh yeah, I can hear myself now. So, good evening everyone. So this is the BioChanges, and this is our third event. We're definitely growing. There's so much more new faces, which is very, very exciting. So just a quick introduction. The idea of BioChanges is to connect engineers, scientists, artists, and different people who are like looking to collaborate and learn something from each other and come up with new projects, new ideas, and to push each other's careers and projects and ideas further. Because we're quite new, please do like us on Facebook and Instagram and this sort of thing. And I think it will also help us all to connect and keep in touch with each other. These are the people that have been supporting us and giving out the talks. So really grateful for that. And now Dr. Laura Alonso will do a quick introduction on how it all started a few years ago. Just a little recap for you to kind of understand where we're coming from, what we stand for. Thank you.
Hello. Hi, everyone. So, yeah, as Oksana said, I'm just going to give you a bit of an introduction of how BioChanges looked like in 2013 and 14. And I'm very pleased to be here tonight because at the time, what we did was basically brainstorm all the time in a very social setting that probably lacked the kind of direction and gravitas to, allow us to execute our ideas. And that has clearly changed in the last 18 months, 2 years, and it's got to where it is today. But, essentially we're after the same kind of thing. We're interested in science, interested in engineering, and trying to, push the boundaries of innovation and also explore the intersection between bioengineering and other fields like the arts and design and that kind of thing. So this slide is a bit of a summary of the people that got together in the beginning. So the BHF Center of Excellence was a big player because a lot of the PhD students that got together during the early phases were studying there in Imperial, of course. Then MIT Media Lab, which, we managed to attract such high-profile innovators early on as our collaborators. The Singularity University, BioCurious Silicon Valley. And these slides is a bit of a, random pictures put together to show you what was going on in the beginning. And there's this picture on the top left showing one of our co-founders, Dominic Southgate, who started a project to develop novel Paralympic equipment that was, just to give you a flavor of some of the work that was going on at that stage. And this, I guess, brings us into last year, 2015, which I think it was a very important year for the growth of the BioChanges community. And I think they've done amazing, to be honest. So the incorporation of members of the Dyson School of Design Engineering and the RCA has been key in making this happen and increasing the impact that changes have today. And that happened late last year, in particular with the initiation of some events like tonight. There's also some other academic institutions like the University of Galway, other initiatives, also academic and startups outside academia that have joined the work that BioChanges are doing. And again, these pictures here just show an example of a workshop that happened last year where people got together to share the ideas. So that's my overview up to today, and I'm just going to hand you over to Oksana. But before I go, I just wanted to say a big thank you to everyone from the second generation of BioChanges for doing this amazing work and putting in all the energy and effort that takes to making this happen and getting people to talk in a room and just network and make change happen. Thank you.
Also a quick reminder of the robotics, who are like one of our key supporters that is doing bioprinting. So anyone else that wants to know more, Gemma is here, and she's happy to talk and discuss any other ideas that you have. And then just like, again, the quick recap from our first event at RCA. That was great beginning, and then also this is the second one at Imperial, which was great, and then the today's with amazing, amazing speakers. So I'll introduce the first speaker. So it's James Field. He's the founder and CEO of LabGenius, a gene synthesized company offering arguably the world's most advanced DNA libraries. LabGenius are able to build highly complex DNA libraries using their own technology in order to produce combinational scanning and rationally designed libraries. So a huge welcome to James.
Yeah, perfect. We have it. Wow. Well, I spent 3 years taking undergraduate biology in this lecture theatre, and I've never seen it so full. So I think that says a lot for the community and how it's grown, and really, really I'd just like to say thank you to the BioChanges group, because bringing this community together in one place is a great achievement. So well done and thank you. So my name's James Field. I'm the CEO and founder of LabGenius. I'm here with my team, just here, Mo, Harry and Ben, just down in this corner here, so please ask some questions afterwards. LabGenius, what do we do? We synthesize synthetic DNA libraries, and we use this for engineering life. I'll sort of get on and how that fits into humanity's story of engineering life. But I'll start just by contemplating DNA for a second. There's a lot of it. 52 billion tons of DNA on this planet. In every single person in this room, we have over 6 billion base pairs of DNA. That's enough to get to the sun 4 times and back. But that aside, DNA is also very crafty. It's a molecule that contains ideas. And that's really what's interesting and unique about it as a molecule. And being able to engineer it means that you can introduce new ideas into life. And the human genome differs from the genomes of other organisms in one way. And that's encoded into it, this idea of do biochanges. It's the idea that means that it's a reason why everybody is here today in this room, because you want to do biochanges. But it's not something that's novel. It's been something that's been going on for thousands of years. And really this started, the first experiment of biochanges, about 20,000 years ago, when humans realized that the gene pool, so all the genes, all the DNA present for a particular species, could be expanded by natural variation, and you could control through breeding which organisms were in particular, which genes were in particular organisms. And so we set about on a grand experiment to engineer an organism, taking something ferocious like the wolf and weaponizing it into something like this. Yeah. Then in the 1950s, we developed a new way to engineer life. People got fed up of waiting for this thing, natural variation, these natural errors in the DNA replication of organisms to introduce new variation into the gene pool. And they started looking for other means in which to grow that gene pool. And in the 1950s, you may have heard of these, they're the atomic gardens. So after the West had dropped 2 atom bombs on Japan, they decided to try and use this technology for a different purpose, for engineering life. So what you see here, the picture on your right, is what's called an atomic garden. So at the center, you have cobalt-60 irradiating lots of plants. So any of the plants that are planted closest to it immediately die. Those a little bit further out get horrific cancer, and the ones a little bit further out from that start to show interesting properties. But trying to engineer life like this is like taking a sledgehammer, hitting an organism, hoping it gets stronger. It's pretty inefficient. And then in the 1980s, I think it was 1983, something happened. This guy in America called Harry Mullis was driving his car along with his girlfriend. And he suddenly had an idea. And that was an idea of how you could take DNA from one organism and transplant it into another using a process called PCR. Unfortunately, his girlfriend dumped him, but he did get the Nobel Prize. So, and in addition, PCR can be used to extend the gene pool as well. So you can use a process called error-prone PCR to target specific mutations within a genome. And it's this technology that inspired the film Jurassic Park. Now, actually, 10 years before Harry Mullis came up with PCR, another technology was invented: artificial gene synthesis. This is the ability. This technology gives you the ability to sort of sit down at a computer and you can design a DNA sequence. You can write down the A's, T's, C's, and G's. And you can send that sequence off to be synthesized, and it will be shipped back to you. And that's a pretty incredible technology. That means the effective gene pool of an organism has just grown from something fairly large to something infinite. Any sequence that you can imagine, you yourselves right now can write down on a computer and send it off, and for about 50 quid, and a couple of weeks later, you'll have it in your hand. And that's really cool because, in theory, any organism can be encoded into DNA. And if you can make any DNA sequence, then in theory, that should mean that any organism that you can imagine can theoretically be made. You're all doubting me now, because if that was true, presumably we would have made things like this, which we clearly haven't. So where's the error in this logic? Well, it turns out that designing novel organisms is pretty tough. So sequence space is this infinite space that encodes all possible organisms that you can imagine. And if you were to pick a point out in sequence space and have that sequence physically made and shipped to you, the chances are it wouldn't encode the viable organism that you're trying to make. And even if you progress through several iterations of this design, the chances are you still haven't made it. And when I started my PhD, we were told that as young synthetic biologists, we would iterate around this design, build, test cycle, and we would build the things that we set out to. And 3 years later, it didn't really work. But synthetic DNA libraries are another way to tackle this problem. So rather than just taking one genetic design or a small number of series of genetic designs and testing them iteratively, you can take many sequences. I'm talking something like 10 to the 12 different sequences. That's more fish. That's more sequences than there are fish in the ocean. And taking that approach means that you can start to build biological systems much faster. It's the equivalent to being able to scan through vast areas of sequence space. And provided you have a good screen for those sequences, you'll be able to pull out the organisms that you're trying to encode. But the applications for this are manifold. We're currently shipping out DNA libraries to people involved in the production of, the green production of chemicals, production of novel pharmaceuticals, bioenergy applications, and agriculture. And our ability to provide researchers with the most advanced synthetic DNA libraries enables them to manufacture novel biological systems much faster. So I've always tried to finish a presentation with an ask, and I'll tonight as well. So our team's growing rapidly, and we're looking for anyone who is a web developer or an R enthusiast. So R is a programming language. So if either of those two things interest you, please come and talk to me after this presentation. And I'd just like to finish by, again, thanking the BioChanges group for the opportunity to come and talk to you tonight. And I look forward to talking to you after the presentation is finished. Thank you. Yeah, we can do some questions.
Well, while it's still fresh, does anyone have any questions? Or otherwise, we'll have a discussion at the end. Well, I guess we can move to the next speaker.
Good evening, everyone. You're living at a really exciting time. We've seen three major revolutions in human history. First, as we started to understand steam and machinery. Then as we started to understand how to leverage electricity and build electrical technologies. Then it went into information technology. Right now is the beginning of the era, though, of biotechnology. We've had exponential increases in our ability to start to read and write the billions of examples of biology that have been put out there by millennia of evolution. We're starting to learn how do we remix this functionality, how we can borrow from the way that one organism can produce an interesting molecule and help transplant that into building entirely new functionality. And there's already teams of designers, of biologists, of chemical engineers that are using this new ability to work with the world of living things, to engineer with it, to imagine a future where, as an example, we don't transplant organs from car crash victims, where we don't try to cure cancer by poisoning an entire patient, and where, as an example, we are able to deal with our growing population pressures with abundance and not considering only a world of scarcity. Underlying all of this is the fact that, as I said, living things are the ultimate remix technology. And so by taking an organism and taking parts of functionality from other ones, you can design genetic interventions which enable entirely new forms of production. So it might be a simple thing like making a pharmaceutical ingredient, or it might be something a lot more fun, like how do you get a steak without a cow, or produce leather to produce cruelty-free handbags. And as I said, teams are already doing this on everything that you see coming out of an animal, everything that you see coming out of a plant, so that we don't crush up tons of plant matter to get a rare perfume, and we don't kill an intelligent being just to harvest what we want out of it. So why do we do this? Well, I myself don't really like to tour abattoirs, don't know about the rest of you, but I'll happily go to a brewery any day. And when you start to think about the resource efficiency, there is no future that gives us abundance that doesn't embrace biotechnology as a manufacturing technology. And it's easy to do. We have 4,400 craft breweries in the United States alone right now, up from almost zero just a decade ago. And not to mention the other ways we are starting to learn how to use biology to intervene in the world around us. The mosquito is the most deadly animal or plant or insect on the planet, with, of course, malaria and dengue fever killing millions, not to mention emergent new pathogens like Zika, causing birth defects across Latin America. And we currently already intervene in our environment by using things like pesticides in a broad spectrum, causing unintended consequence, as opposed to using technologies like Oxitex, where we can elegantly target only female mosquitoes that bite, spreading these diseases, by building mosquitoes that only transmit male offspring, thus being able to target just the problem, not the broader effect. And it's also being able to do things like build living vaccines. The work of a UK-based company, Proparium, has been on how you can engineer a bacteria which can express on its surface a plurality of potential vaccines, which brings things like a vaccine to the common cold a potential reality, and also means that you can start talking about updating a person's immune system, not just off of a single pathogen, but this is the sort of thing that can be done on a regular basis. Not to mention, as I said, the issues we face in terms of resource abundance. With today's technology for farming, with the growing population of 10 billion that we're facing, we've got a 60% gap in calorie production. And that's ignoring the fact that we're also undergoing a major transition in how we make things in the world. We're going from burning the previous biological feedstocks, like oil, that have been built up over millennia, to how we can produce renewable inputs to all the chemicals that we use in our daily lives. So it's clear that our ability to make renewable products is going to be paramount going into this century. The problem is, with all of this demand for what we can do with biology, our ability to work with it today is still pretty bad. Inflation adjusted per $1 billion spent is actually an anti-exponential to bring new drugs to market. It's sarcastically called Eroom's law. And that's because at its heart, working with biology is hard. These are complex systems. There's lots of things going on in them. And it's unfortunately not a problem only of coming into medical interventions. It's still over a decade on average to bring a new crop trait to market. And it's still 7.5 years to bring a new industrial biotechnology process, making things like the rubber that goes into the tires of your cars or the soles of your shoes. Now, at Synbase, we're focused on how do we become more productive working with biology. And I mean that in every sense of the word. It's how do we compress the time cycles to do research in the first place? How do we build manufacturing processes that are more efficient in their outputs per unit time? And ultimately, how do we get to a world where we are making with biology in a reliable and cost-effective way? To do that, we've been paying attention to how we've already addressed this sort of exponential complexity before. One of the best such examples is the computing industry, where everyone is familiar with how Moore's law has meant an exponential increase in how much can get done on your CPUs. It's why your iPhone is more powerful than the crazy supercomputers of the past. What people don't always know, though, is that was through a stair-step of innovations. And one of the most important ones was back in the early 1980s, where we finally introduced high-level languages for describing what a chip should do, Verilog and VHDL. This meant that up to then, what had been an artisanal discipline, where a semiconductor was something laid out by hand, designed by hand, and thought about by hand, became something that could manage the exponential increase in complexity, such as the 2.6 billion transistors that go into a modern processor. It meant that we could build tools that help us engage with this problem space, and it wasn't limited purely by our own imagination. So at Synbase, we've introduced ANTHA, which is a high-level language for describing how you can work with biology. It means that we are building this stack of technologies, these EM tools that mean computers and robots can conduct experiments, and software can support your ability to think through this complexity. And it means that you can do pragmatic things like investigate all of the complexity that comes into play with biomanufacturing. So in this case, talking about how we could investigate dozens of different things that could be influencing how you could make with biology for a major therapeutic production platform. And we discovered that you can actually understand this, even though there's complexity to it. You can unpick the fact that there's cross reactions between genetics and environment. But it's about structuring the way you search this. It means building a map on how these systems work. And it means not just randomly screening everything out there, where, as an example, I would probably pick what was the highest yield. It's understanding the reactions and interactions that go on in these complex systems. So I know I instead needed to pick the system, genetically speaking, that was robust to temperature. So when this thing was manufactured in 50,000 liter fermenters with no control over temperature, it still works. And as a point of comparison, not rationally investigating, not searching in parallel, not building models of how these systems work, would be like trying to find Earth in the Liopas supercluster, just one galaxy at a time. We're there. There's a lot of them. It also means changing the way we work with biology. If any of you have worked in wet labs today, it's a laborious process. We're still thinking about this problem by hand. We're trying to design our experiments on a piece of paper and calculate them. We're trying to engage with the equivalent of building an i7 processor with, a spreadsheet and a calculator. It also means that we're trying to take people that have spent a decade of their lives learning these skills, learning these complex systems, to act like glorified robots. By introducing a high-level language means all of a sudden it's robots doing that. It's software taking that grunt work out. And it's opening it up for you to focus on the question of what are you trying to design. We're doing this by talking to all of the different equipment that make up laboratories. We're doing this by building tools that let you engage with all of these unknown questions, that help you understand what a genetic intervention will do, that help you build tools to understand how these changes will work in the real world. And when you start talking about the incredibly complex bits of kit that are being built into the future to enable things like parallel bioreactor arrays to rapidly manufacture products in, say, an emergent outbreak, you realize you have to talk about designing with biology being something that can be done on a computer in 45 seconds. Like in this case, where an entire port designed for an intervention to an organism can be simulated Everything that you need to do it can be computed for you. And ultimately, a robot can 3D print that organism with you walking away after only a minute of work. If helping to make biology something that we can indeed engineer with is of interest to you, I encourage you to take a look at ANTHA. It's open source up on GitHub. And if you have any interest in the molecular biology side or the computational side, we're hiring actively and are just across town. Thank you very much.
Great talk. Just a quick reminder, where you're sitting, there's little slips where I would like to hear your feedback, your ideas, and would like in the future to connect and build a big community for all of us. And now Tony, who's part of the BioChanges, will introduce the next speaker.
All right, so our next speaker of the evening is RCA alumna Rachel Wingfield. Rachel founded Loop PH in 2003. It's a spatial laboratory which works cross-disciplinarily, merging the areas of design, architecture, and the sciences. This experimental studio dreams and speculates on prospective scenarios in order to envisage the impact of emerging technological and biological futures. Thank you.
Thank you very much.
Hello, hello. Hi there. Wow, there's loads of you. It's amazing. So I'm going to give you a designer's perspective and talk a little bit about the work that I've been doing over the past like 14, 15 years, which looks to merge biology, design, and basically move in between lots of different disciplines. So thanks for the invite. As you described, we have this studio that's in northeast London, but you're welcome, not all at the same time, to come and visit us. We call it a spatial laboratory because we sit between so many different disciplines. It's kind of hard to describe exactly what we're doing, but we kind of see ourselves as interfacing architecture, the life sciences, and art, and basically developing participatory programs that deal with public engagement. Oh, my picture's gone. So basically, I'm a designer, which means I've got like 300 slides. I'm going to have a freak out if you can't have a visual very soon. Yeah, it's beautiful over here. So yeah, I mean, I guess originally, you could call myself as an artist. I work incredibly transdisciplinary. Simply can't speak without picture. I can just hold it like this. Sure, but it's like 10 years. Oh, okay. Yeah.
So no matter how much you practice, there's always some unexpected surprises. So while we're sorting this out, which I'm sure we can do that, Amanda will introduce the next speaker. Yeah, so now we welcome the lovely Anna Dimitriou. She's a visual and performance artist, creating beautiful and thought-provoking fusions between arts, craft, technology, and the life sciences. And amongst a lot of things, she explores and communicates the shifting relationships between us and our unseen microbial companions. And she's currently an artist in residence at the University of Oxford on the Modernizing Medical Microbiology project. And thank you.
Hello. Just checking how I should use this. Right, well, very nice to be here. So I'm an artist. I don't have a startup company, and I'm not actually hiring anyone. So I don't even really need any interns. But you know, if you're really keen. So yeah, I'm an artist, and I'm kind of obsessed with bacteria. That's probably the best way to put it. So I spend almost my entire time thinking about bacteria and playing with bacteria in various laboratories, and I work with kind of craft techniques and various things, and I'll go into that. This piece that you can see in the picture is my effective textiles chest from quite a few years ago, and this was kind of the first piece where I embedded textiles into agar and grew bacteria on them. This was made from environmental bacteria that were swabbed from around the city that I was working in, and the little jars are they're kind of natural dyes embedded with different silks, embroidery silks, and different things like turmeric and safflower and madder root, which are all antimicrobial. So I'm very obsessed with the relationship of dye to microbiology and the process of microbiology. And then based on that, I developed this piece, which is my MRSA quilt. So MRSA, if you don't know, if there are any Americans in the audience, MRSA, that's what they call it. It's methicillin-resistant Staphylococcus aureus. So Staphylococcus aureus is this very popular bacteria. At least 30% of you are carrying it on your body now. It likes sort of warm, kind of sweaty places. It likes the front of your nose. It likes your armpits, and it likes sort of the nether regions as well. And it just lives on you normally and happily. And at some point, sometimes it becomes infectious, and they don't know what switches that. They're studying that now. What switches it from being this thing that lives on you to killing someone? This image was actually used to illustrate a paper in the Proceedings of the National Academy of Sciences, which was about a paper where a patient, well, somebody who was participating in a research project was being studied over time for their carriage for Staphylococcus aureus, and then all of a sudden they died of Staph aureus infection, and they tracked through genomics the process of change through that, but it's still not very well understood. How does it change? So this piece is methicillin-resistant Staph aureus. The bacterium evolved to be methicillin-resistant in 1960. So it's a drug resistant. So a lot of bacteria, and this is a really increasing and serious problem for us now, a lot of bacteria are developing drug resistances. So these antibiotics that have been with us since the 40s are now stopping working. And we could be in a situation in the near future, if something doesn't happen, hopefully something will, but if something doesn't happen, where antibiotics stop working, and a lot of infectious diseases we thought we cured are going to come back and haunt us. Particular dangers are these sort of super bugs, these ones that have become multi-drug resistant, and things like extensively drug-resistant tuberculosis. Tuberculosis is a disease that currently infects 1/3 of the world's population. So if it becomes extensively drug-resistant, in a widespread form, that's going to be incredibly dangerous. So this piece was made using the actual bacterium. So the blue is the MRSA bacteria, and it was embedded onto chromogenic agar plates, which are color-changing agar plates that change to a color of blue in the presence of MRSA. So the bacteria grows blue on it. So I impregnated the material with it, and the bacteria grew into it. The patterns are created using different tools and techniques in the treatment and diagnosis of the disease. So these sort of polka dots here, these quite cheerful looking polka dots, they're actually vancomycin susceptibility discs that they use in hospital laboratories to test whether the bacterium is susceptible to that antibiotic. And it is in the case of this, because it's methicillin resistant but not vancomycin resistant. The stripe is a quercetin strip. Up the top there might be of interest. That's an embroidery with turmeric, and you can see that there's just a small zone of inhibition around it. So the MRSA bacteria hasn't grown right up to the turmeric. So it's obviously doing something that's antimicrobial. People say, oh, there's all these things, you know, turmeric's the new wonder drug, or this is the new wonder drug, or this is the new wonder drug. The antibiotics that we use are very, very powerful, and what they do is they study and find out what the active molecule is, what the specific thing is in that, and use that to kind of synthesize the new antibiotic. So turmeric has something, but we need to look more into it, perhaps. And I do participatory workshops. So this was done at the DNA, and we did workshops where like hundreds of people came, and they made their own MRSA quilt squares. So they brought things that they thought were antimicrobial, and we tried them out against the MRSA. And we also had lots of things there available for it. We had my collection of historic antibiotics that goes back to neosalvarsan at the moment, which is 1912. It was invented by Paul Ehrlich as the first treatment for syphilis. And I've got Prontosil as well, which was 1935. That's synthesized from a red azo dye by Gerhard Domack. And what was lovely was an older lady came to the workshop that we were running in the VNA, and she told us the story when she was a tiny child and she was treated in hospital with Prontosil. So it's fascinating to collect these stories from people, and I think the storytelling and the story sharing, because I always run the workshops that I do with the scientists I'm working with too, but what it does is it breaks down the hierarchy between the participants in the workshop and the scientists. Because normally if you go up to a scientist, if you're not a scientist, it's very difficult to kind of just ask a casual question to them, because you feel like an idiot, or they'll start saying something really complicated and you can't get the point across, and you just, that doesn't happen, and they don't get the chance to talk to just everyday people. So by making an artwork together, this breaks down all the barriers. So I work with a lot of leading scientists to kind of run these workshops. Just a note, we took the plates back to the lab to inoculate them with MRSA, in case you were worried that we had MRSA bacteria at the VNA. We didn't. Somebody may have, but we didn't. This is a piece from my project which was called The Romantic Disease, and it was about the history of tuberculosis and looking at the past treatments of it and the future direction of it. And so this piece was called Rest, 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 and it was about how basically before antibiotics, the only treatment that we had for a tuberculosis patient was to tell them to rest. And there are these entire myths created about why they should rest, and how they should rest. And so patients with tuberculosis, I'll give you an example. The Craggy Nos Sanatorium in Wales, just before the war, the children that were kept there, the sickest patients would live on the balcony of the hospital in Wales, and they would live there year round. So they would sleep with tarpaulins on their beds, so to keep them waterproof. And in the morning, the nurses would come and shake the snow off the tarpaulins where they were sleeping, off the balcony, because they believed that fresh air was the ideal cure for TB. I mean, it's good to have airflow. The main risk factor for catching tuberculosis is to sleep in a confined space with somebody else with tuberculosis. But the idea that you needed to be in this kind of freezing environment. So patients would be kept outside all the time. And some of the patients were kind of were actually immobilized as well, because they took this rest idea to the extreme, and they would make plaster casts of the children's bodies and strap them to the plaster casts, and sometimes for 2 years. So the treatments were bizarre and extreme. And this is probably one of the most extreme, pneumothorax treatment, artificial pneumothorax. So what they would do would be to collapse the lungs of the patients and suck out the air to give the lung a rest. And this is I quite often work with historical objects. I'm kind of obsessed with the history of microbiology and use them in my work. So this is an artificial pneumothorax machine that I've carved and engraved. So the carving is based on the granulomas that form in the lung and the cavities that form in the lung. And I studied that by going to handle lungs in the dissection room to kind of look at the tissue, the way it grows and things, and squeeze them. Actually, healthy lungs were like cosmetic sponges, which is quite an interesting thing. And then the unhealthy ones are like sort of tissue paper, it's sort of like soggy tissue, and don't reinflate. So it's a really weird experience. And you can see these, the needles there, which are actually still in the machine, and they were the needles that were plunged into the lungs of the patients. The cylinder also has the engraving of what they describe as the characteristic scattered red ribbons that you see under the microscope. So if the way they stain tuberculosis, which was the original staining technique was discovered by Ehrlich, that invented the Salvarsan. It looks like red ribbons that have just been thrown down. It's really hard to find the tuberculosis bacteria on microscope slides. So it's a really inefficient way of diagnosing it as well. And that's a close-up of that. This piece is called Where There's Dust, There's Danger. In 1912, the British Society for the Prevention of Consumption published that where there's dust, there's danger. They said that the primary cause of TB was dust, and that you'll notice it when you go to buildings like modernist architecture. Actually, people don't realise a lot of it stems from tuberculosis prevention ideas and myths quite often. So like the staircases, when you see buildings with staircases with gaps down the side, that was so dust wouldn't collect at the sides, because they thought that the dust would cause it. You can't actually catch TB from dust. What they thought was people would spit and the sputum would kind of dry, and then when it was swept up you would get the dust in your lungs. You can't catch it like that. You need somebody to cough on you with active tuberculosis. The weird thing is they can have multiple kinds of active tuberculosis. So somebody could cough on you, one lung could have one form of tuberculosis, one lung could have another. Sometimes it's active, sometimes it's not. And then this piece was impregnated, so it's made of needle felt, and you can see the images of the kind of the lungs that have been collapsed, and also the granuloma, these sort of walled off areas of the where the bacteria is walled off by our immune system, because it can't be killed by our immune system. All our immune system can do is wall it off, which is why there was a resurgence in the disease with HIV, because your immune system is what keeps it at bay. But when you have a weakened immune system, it's much more difficult. And then, so the piece is made of needle felt combined with dust, and they're impregnated with the extracted DNA of tuberculosis. So they actually have this tainting of the disease, which is something that's really strange to me, and it's in all the historical objects that I collect, that these objects, these books I collect, they've all been handled by people who have been through this, which is kind of strange to me. Yeah, this is about the kind of, it's a kind of tiny modernist furniture set, sort of model of furniture that's impregnated with Promethazine, the antibiotic that was isolated from a red azo dye, and it's called The Consultation, and it's about the idea of actually they discovered that the earliest antibiotics were not working almost immediately, so every antibiotic had early drug resistance, so that's why things like combination therapies and improvements in kind of drug production over time. I'm doing a residency at the moment with the University of York, looking at the hunt for new antimicrobials as well. This piece was made in collaboration with the University of Leeds, and it's about another treatment, which is human fecal transplants. Clostridium difficile bacteria is really fascinating because it wasn't a problem for us. It's something that little children carry. It was no problem whatsoever for us, not causing any illness. And as a consequence of antibiotic use, it became a problem. So it would be just one of the many bacteria living in our kind of gut. And if you take broad spectrum antibiotics, certain kinds are more attuned to getting it, then what will happen is it will kill off the diversity of bacteria in the gut, and that leaves a space for this bacterium to grow and kind of proliferate. And once it's there, it's drug resistant, and it's really difficult to kill. So what they've discovered is that one of the effective treatments for this, the other alternative is just more antibiotics and more antibiotics, which creates a vicious circle. The efficient method is human fecal transplant. So you actually take the gut bacteria, poop sample from a healthy person, you sterilize it, you kind of pulverize it, you mash it up. I went through the whole process of doing this in the lab. You mash it all up, you kind of strain it through, they use these the sort of strains that they use in brewing. If you've ever brewed beer, they buy the same thing. And you strain it and you get this like liquidy form of it, and that is put into a patient. I can go into the details if you want later, but and in 90% of cases, after a maximum of 2 treatments, the patient is cured of C. diff infection. So it's very effective, but obviously it's not suitable for everyone, because patients with bowel cancer, for instance, or patients with Alzheimer's disease and things, it's not going to be very effective for them. You have to keep still. It's quite an invasive treatment. So I worked with the scientists to make that. So this piece is currently it's on permanent display at Eden Project in Cornwall. The carving is the images of C. diff spores, which they think are the cause of reinfection. Spores live in the biofilms in the gut, and even if you kill off all the bacteria using antibiotics, later you move about or something, biofilm breaks down, the spores are released, and you get a reinfection. The intestine there contains the sterilized human fecal transplant. I had to sterilize it because otherwise it would possibly explode in the gallery, and you don't want that. And the other piece is tubing that's used in their gut model. They use to study the bacteria, and it's coated with different materials that have been embedded with different gut bacteria. So there's C. diff, Enterococcus, E. coli, different bacteria, all on that. And also work with kind of performances as well. This is my hypersimbient enhancement salon, where I kind of did consultations for people about ways that they could enhance and improve their own microbiomes. For instance, TB has in the past been described as benefiting creativity. I don't know if any of you have studied Keats, but he was well known to be infected by TB, and there are a lot of papers written on the idea that he wasn't actually very creative until he got TB. So there's lots of other things around it. So I was maybe hypothesizing that this could lead to it, but there's already kind of suggestions that the changes in your gut bacteria can lead to other outcomes. So they've grown mice in sterile conditions and inoculated them with the gut bacteria of obese people, and the mice have become obese. They've also created anxious mice by giving them the gut bacteria of other anxious mice or people, I'm not sure, but anyway. But I was kind of giving consultations around this. It was a playful piece. And then I was approached by Paloma. I think you're here, aren't you? I saw you earlier. From the UCL iGEM team last year, and I developed the piece because they actually genetically enhanced the bacteria to create serotonin, which reduces anxiety. So they've made an E. coli, probiotic E. coli that you could drink in theory that would make you happier. And so it's got problems though at the moment, because it's on a plasmid that codes for chloramphenicol resistance. So it needs to be kept in the presence of chloramphenicol and quite a nasty antibiotic for it to grow. I'm sure all these things could be dealt with. There's the issue is you don't know how much it's going to grow as well and how much it will proliferate. You might have a kind of C. diff-like problem. So research needs to be done, but it's interesting that this is coming up. So we have these jellies that you could eat, and they've actually got chloramphenicol in. So definitely it would survive in there. There's an outside chance you might sustain some nasty side effects. But the lipstick, again, that's freeze dried with the bacteria. That so that's usually the best way of kind of having a bacterium and keeping it in order to kind of eat it like a probiotic. 'Cause these yogurty things are not necessary. It's not a very good way of doing it. They're not surviving in everyone. And they were a food supplement in the little vials there. And my most recent project's been about whole genome sequencing. I sequenced the entire genome of the Staphylococcus aureus that lives on my body, in my nose actually, left and right nostril. I've got 2 different species. And this dress that I made, at least 2 different species, probably more actually, is impregnated with that Staph aureus, with MRSA and with BRSA, that nasty vancomycin resistant one, which is kind of resistant to our last ditch antibiotics in that. And I took the output of the whole genome sequence. I worked on it with Alex May, who's here, and that was video mapped back onto the dress. So it's actually a journey through the light output of the whole genome sequence of the bacteria from my body, and then the data is projected behind. And then finally, we created a virtual reality experience. So you can actually kind of walk through the data and be inside it, be inside the bacteria from my body and hear its story. It's resistant to 3 different drugs. It's got the BLAZ gene, which means it's resistant to penicillin, the NORA gene, which means it's resistant to ciprofloxacin, and the TET38 gene, so it's resistant to tetracycline. But it's not methicillin resistant at the moment. And that's on my website, so that's it. Thank you.
Yep, okay. It is now my great pleasure, just a slight change of schedule, but we are going to have the amazing Professor Oren Katz and Dr. Ayan Abzar going to come and speak to us today. Incredible artists have just started as visiting professors at the Royal College of Art as well, for many of you. As bio artists, their work at Symbiotica, the University of Western Australia, and the Tissue Culture and Art Project are incredible examples of the exploration of tissue engineering and regenerative biology technologies in looking between and exploring between the areas of art and science. They speculate, they question, they look at the future developments of biotechnology, and we are very happy that they are here tonight to speak to us. Thank you very much.
Hopefully technology would work. Let's see where we are. Oh, okay. So, yeah, just bear with us for 1 second. Now, this is a great example, actually, what we've seen just before. Just after the discussion about rationally designing living creatures and the bright future that we're facing, even a fucking projector couldn't work. Yeah? So just put it in context.
Only on this one.
All right, so yeah, thanks so much for inviting us. It's really great to be here. Both Renate and myself, as you heard, just moved here to London on a partial appointment at the Royal College of Arts. As you can see, my title is a visiting professor of contestable design, and this is what I'll try to do with you tonight. I want to put forward some contestable ideas. We're not techno-utopians. We work with the technology of life because we want to understand it better, because we want to figure out where we are. And I'm also, as you heard and as you can see there, a director of Symbiotica. So, and Jonati is our academic coordinator. We're not recruiting employees, but we might recruit some students, if you like what we have to say here. So, in a sense, what we've seen already this evening is the realization that technology is becoming more lifelike, and at the very same time, life is becoming more technology-like. There's this strange meeting point that we see happening in front of our very eyes that we feel as artists need a lot of cultural scrutiny. We're trying at the very same time to relinquish control over the technology that we developed. Think about killer drones, for example. At the very same time that we're trying to assert our control over living biological systems. So this is really kind of the premise of the type of work that Jonati and myself have been doing for the last 20 years. And 16 years ago, we set up a laboratory which was at the time the first of its kind in the world, in the sense that it's an artistic laboratory dedicated to the learning, research, and critique of the life sciences through experiential engagement. We were very, very lucky that the University of Western Australia actually allowed us to come into a science school, the School of Anatomy, Physiology, and Human Biology, and allow us to set up our research lab and set it up in such a way that it is a lab like any other lab in the school. The only difference is that we research life and how our relationship to life is changing and shifting in the light of what's going on, obviously with current modes of technology and knowledge, and allow us to be there and do the work in a very hands-on way. So we were very interested in researching and identify those places where we feel needs the most cultural scrutiny. So we are basically looking at trends in contemporary science and technology to see where we feel we need to pay more attention. We explore the possibilities of those new technologies in order to propose alternative directions and create objects for cultural debate, engage in what we refer to as those contestable objects. We're not interested in finding the cure for cancer. We're not interested in trying to feed the world. We're interested in engaging with those very important ontological breaches and ontological holes that people like yourself are digging for us, and how we as a culture would either dig ourselves out of it, or at least be able to articulate it in a way that will make sense for the rest of us. So we also, but as you heard today, what biotechnology, what these new trends in the way we apply our knowledge of life allows us is a new, basically, set of tools and a new palette of artistic possibilities. So regardless of what we think about it, the fact that life is becoming a raw material for us to be engineered, it also provides artists and designers with a totally new palette of possibilities in order to be able to engage with them. The way we do it in Symbiotica, we're not speculating about this new palette, we're actually engaging with it in this experiential way. We are dealing with the actualities of what's happening now and here in the context of those new tools that are being offered to us by engineers and scientists. We also research and develop strategies of what it means to start to take those kind of things into the cultural realm. So I'm sure that Anna would agree with us that trying to put something like that within the context of a museum and art galleries, which were designed for the last at least 150 years to be the graveyards of everything. You know, those places are designed to keep things dead and dead as much as possible for as long as possible without any changes. We come in and we try to put living systems within those confines of the galleries and the museums, within the cultural context. That raises quite a lot of very interesting encounters. And so we are trying to develop those new strategies and those new tools of displaying living biological systems within a cultural context. And obviously we feel that there's a really important part of reflecting and engaging with those moments of uncomfort, those zones of uncomfort that some of us feel, and as the years go by, less and less of us feel uncomfortable in regard to this manipulation of life, but there's still zones of uncomfort, those ethical issues that we feel needs this cultural attention. So at Symbiotica, the way we've done it, as I mentioned, it's a research lab, but we're also an academic program. So we run what we think is the most successful MBA, the Masters of Biological Arts, and the most useful one as well in that. We run exhibitions, We have a residency program, so we open to artists and designers and actually anyone else who wants to come and engage in a very experiential way with the tools of contemporary biology to come and spend time with us and do research residencies. And then we organize symposiums, seminars, and workshops, hands-on workshops. Now, we also develop tools for artistic research and display of living biological systems. So here you see a work of Matt Johnson, who was an RCA graduate who came to do an internship with us, working with Alicia King, an artist who was interested in setting up a bioreactor to show living biological tissue for long durations of time for the gallery. So those are the sketches, that's the actualities. If we would actually try to buy a machine that would do a similar thing, it wouldn't be to the specification of the artist to start with, but also it would cost us at least 20, if not 50 times more than what it cost Alicia and Matt to develop this bioreactor here. As I mentioned, we see ourselves as a research center that deals with the question of life and how our relationship to life is changing and shifting. So we are looking at all gradients of life, from the submolecular, if you like, all the way to the ecological. And this is just a very rough taxonomy of some of the areas of research that have already been explored at Symbiotica. But the main interest of United and myself for the best part of the last 20 years was tissue engineering. We realized that something really interesting happening within this realm, and we decided to explore it further. And we just recently, I curated a show with a couple of collaborators, looking at the 20th anniversary of the first public appearance of the mouse with the human ear growing from its back. So looking at the history of that, how the popular culture and how artists responded to this image. Now, kind of a really interesting piece of trivia, how many people you think have ears that have grown in such a way of tissue engineering? One, as far as we know, and that's not even published in a peer-reviewed journal, so it might be no one. Now, the interesting thing of tissue engineering is more than the actual technology and how it, if it's effective or not, it's the type of conceptual shift that change our relationship to our bodies, change the relationship to how we see the body being repaired and fixed, and how plastic the body is. In a sense, this is what the mouse with the ear spec generated. It brought the surrealist dream alive. And it was brought alive by the hands of scientists and not artists. In my interviews with them, they were a bit reluctant to talk about the art historical references that they were using in order to come up with this object, but it obviously existed within our psyche, the idea of this chimera. So here we see this kind of monster comes alive, the body is becoming plastic, but even more than that, our imagining of how the body would be repaired went through a major shift. This slide is actually from a presentation by Professor Joseph Vacanti, who was one of the founders of the field of tissue engineering, and that's from a presentation that he gave back in the year 2000 at the second or third International Tissue Engineering meeting in, interesting enough, Disney World in Orlando. So here you see, this is how we imagine the body to be repaired in the 1989. Yeah. So the idea there was that you would replace any failing or ailing or missing body part with some kind of a hard mechanical apparatus. Yeah. So what we can refer to as the Tin Man approach. Yeah. So bit by bit, you would replace the body with those hard mechanical engineering objects that would come instead of those soft biological tissues and organs. Only 10 years later, 1999, you see the very same idea of fixing the body, but now it's the body fixing itself. It's trying to harness those latent regenerative powers of the body in order to replace those missing, ailing, or defective body parts. Now, in 1995, when the whole story really exploded, an international consortium of scientists was set up to try and grow a heart using those technologies. So for me, doing design at the time, I was really interested in kind of reversing this logic. So if in 1989, we were told we can replace the heart with a pump, in 1995, we were told we can grow a heart, my question was, why not grow pumps? Why not use those tissue engineering technologies in order to generate functional tissue products that would never be reintroduced into the body? But I figured that actually this is raising so much more interesting philosophical and ontological questions than trying to develop products. that I decided to pursue this project as an artist, and I invited Jonat to come and join me to what we then called the Tissue Culture and Art Project. And actually, this is my original thesis from 1996, where I was looking at the idea of living surfaces, and I suppose it was what can be called now a speculative design project, because at the time, I'd never been to the lab. And only with this idea of being able to do something with it, I was able to approach a scientist who, to our surprise, invited us to come and work in her lab, and that's when we started to actually grow living tissues as art.
So since 2000, we've been growing, actually hands-on growing tissue, using tissue engineering technique to grow. It started with growing sculptures, if you want. And we even worked at the, in Boston, in the tissue engineering organ fabrication, Dr. Bacanti lab, to grow all sorts of sculptures. And later on, we talk about it more. But the first sculptures that we grew, and actually were able to take it out to the public as a visceral thing, not just as a photograph or as a non-living object, was what we refer to as the semi-living worry dolls. And I don't know how many of you know about the worry dolls. Those are little crafted dolls that children put under the pillow, and they can tell their worries to the dolls. So we were invited to present those dolls in a gallery setting, and of course, you know, you need to look after those dolls, you need to create a whole infrastructure to actually grow the dolls. So we built a lab that you can see over here with the sterile hood, with everything you need to look after those two tiny worry dolls that you can see here. Actually, we had 7, but you see example of 2 of those here. And they were grown inside a bioreactor that you can see here. Those are the red vessels, and over the length of the exhibition. And unlike computer and everything and things like that, but we compared biology to, of course, the digital technology. Unfortunately, you do have to look after them. You do have to feed them. You do have to change their nutrient media. And things can and usually do get wrong, as you can see here. So what we did here, actually, we did a feeding time when people could come and actually see us feeding those semi-living worry dolls, like, you know, the way you feed animals in the zoo. Again, recreating and emphasizing the thing that when you work with living material as a technology, you do work with a material that some people may think is slightly different and needs different consideration. And also, of course, we were very interested in what people feel about presenting for the first time living, semi-living tissue construct, and it was in Austria, a place that associated a lot with eugenics, we all know. I mean, England as well, but we can talk about it later. And we wanted to see what people will think about this kind of thing. And we had a computer. You can see it was a long time ago. It's an old computer. And people could actually write to those semi-living dolls their worries. And we were sure that the worries will be a lot about biotechnology and, you know, how artists can actually play with life. But in some ways, then those worries become very personal. And some of the people actually treated them almost like a voodoo dolls. Again, I think the fact that they have living tissue inside them, they were actually living visceral sculpture in the gallery, created this kind of relationship with the audience. Should I think about that? Okay, so as I said, again, when you want to grow something as tiny as that is definitely quite humble, you have to build a whole infrastructure around that. You have to build the lab. And this lab was built in 2011 in the Science Gallery in Dublin, and it served many artworks that had different kinds of light inside them. And you can see on the left again, the worry dolls over here. And this time we actually put a microphone so people can whisper the worries to the doll to see whether it affects the growth and the condition, as well as other works around here that maybe you want to discuss. We have to hurry. Okay. and here you see them. Oh, and here, of course, by the end of an exhibition, no one talks about the idea that, you know, when you turn off a computer, you can turn it off on the next day. When you turn off light, that's it, that's for sure, that's the end. So we also discussed the fact that our art in the end of an exhibition must be killed. And we try to make the audience actually part of that. Because just by coming and watching this art, you are part of the whole process of making art into frivolous artwork. So this is the ritual of killing, where people come and they kill our artwork by touching them.
Yeah, so we had to develop a new language, really, because a lot of what we were doing was something that was never described culturally before. A lot of the ethos, and this is what we talked about before, is based on the fact that we don't have a cultural language to engage with those ontological holes, with those issues that we need to deal with as we are starting to use life as a raw material to be engineered. So after the War Eagles, we were actually invited for a very celebratory exhibition that was supposed to celebrate the so-called completion of the working draft of the Human Genome Project. And our response was to deal with the hype around the Human Genome Project. Some of you might remember there was an explosion of those extreme unrealistic statements in regard to what would come out once the Human Genome Project is complete. And the interesting thing is that actually those celebrations in June 2000 for the completion of the so-called working draft of the Human Genome Project were more political than scientific. The Human Genome Project wasn't even close to be completed, but there were some political issues with the competition between the private Human Genome Project run by Celera and the public Human Genome Project that was heavily invested by governments and the public. So there was a need to get the media off the back of this story, and therefore there was this non-scientific celebration of the so-called completion, which never really occurred until 3 years later. So anyway, we decided and we proposed a project that was looking at this idea that if the impossible is now possible, as some of the statements declared, pigs would fly. And if they would fly, we wanted to see what shape those wings would take. So we looked at the three evolutionary solution of flight invertebrates, the birds, the bats, and the pterodoxals, and we decided to grow them out of pig tissue. And now we also looked at this idea of the iconography of wings in Western art, where usually bat wings are attached to devils and demons, bird wings are attached to angels, and those pterodoxal wings are not attached to anything within our iconography because we just found out about them late in our history. And in a sense, we can treat it as a way of how we're going to engage with this new knowledge. It can either destroy us, it can save us, or we can say, just wait a minute, we really need to figure out what's going on here. And so we color-code them according to those types of assumptions. We harvest bone marrow stem cells from a knee of a pig, those mesenchymal stem cells, that we then design the wings using three-dimensional software. So in a sense, that was one of the very first 3D printed tissue constructs. And we're doing it as we were doing this residency in Harvard. So we also introduced this CAD-CAM protocol for the tissue engineering and organ fabrication lab in Harvard, growing pig wings. And those wings have grown, we've grown them for about 9 months in those microgravity bioreactors. We got almost complete completion or complete replacement of the synthetic polymer with the live bone tissue that we grown. And then we pull them out and put them in those cheap jewelry boxes, coated them in gold. Now, in order to develop a new language, we also had to realize that a lot of what's going on within the context of contemporary biology is really dealing with what we refer to as the aesthetics of disappointment. People who would come to the gallery, especially after the hype of the Human Genome Project, would expect them to see, if not flying pigs, at least wings which are big enough for pigs to fly with. Now, those were one of the biggest tissue-engineered constructs to be grown to the date. That was 15 years ago, 16 years ago, but they were tiny. The wingspan was about 4 centimeters. The thickness was about 4 millimeters. So people would come and see those wings in those cheap jewelry boxes and get quite disappointed. So after doing those kind of symbolic gestures, we decided to move towards what we refer to as those pseudo-utilitarian projects, where we imagine, okay, what you can do with tissue engineering if you're not going to introduce it back into the body. And the very first project we've done in the series of what we call the technologically mediated victimless utopia was our in vitro meat project. So back in the year 2000, we worked in the lab where they were working with in utero tissue engineering. One of the scientists was harvesting satellite cells, so those progenitor muscle cells, from a lamb that wasn't even born. And she had so many of those cells that she didn't know what to do with them. They basically clogged all of the incubators in the lab, and we decided, okay, bring it to us, we'll grow meat out of it. And that's how our project of in vitro meat started. But we also were very, very interested in this relationship that we form with other life forms. So when we start to think about biology as engineering, when we start to think about life as a raw material, the most intimate relationship we can have with those life forms, more than sex, is to eat them. Because we incorporate them into our body, they're becoming part of us. And we started to look at it in this way. So here's a short video of how it was done. This video is for many years later, but it kind of follows a similar trajectory. Because of sound. Think about it. Anyway, this is one of the steaks we've grown. Again, aesthetics of disappointment. We were able to grow about 5 grams of that meat in each batch of growth that we've done. And we would basically isolate cells, harvest cells from the animal, directly from the animal as primary cells. We would then isolate those satellite cells and culture them in the lab in those microgravity bioreactors for about 3 months in order to get this piece of meat that you've seen before. Now, the piece that you've seen before, we couldn't really eat because the lab didn't have the license to grow food. So the first time we were able to actually grow and eat a piece of meat was in 2003, where we were invited to show our work in an exhibition in France called L'Art Biotech, where we were able to construct a fully functioning lab with a dining room in the gallery. And we were playing on what constitutes foul food. And we knew that the French people don't like the idea of engineered food very much. And most other people don't like the idea of eating frogs very much. So we decided to tissue engineer frog steaks and use them as the first ever in vitro meat to be grown and to be eaten by humans in a cultural context or anywhere else, if you like. So here's some footage from the original performance. So for the 3 months of the exhibition, we would come every day and care for our steaks and feed them and replace the nutrient solution. And at the very last day of the show, and actually here you can see as well, what we've done was to release 2 frogs from the edible frog distributor in the town. And they lived in the gallery throughout the show, looking at us, feeding those decontextualized parts of the frog body. And we then, at the very last day of the exhibition, we had this ultimate nouveau cuisine dinner where we basically served this in vitro meat to people from the audience and the curator of the show, and obviously ourselves. It wasn't very tasty. Worse than that, the texture was horrible. The problem we had was that the polymer, because we had the confines of the time span of the exhibition, the polymer didn't degrade completely, and we're using PGA mesh, which was like felt. And we didn't exercise the muscle cells, so they were like jelly. So we ended up tasting like something, or the texture was very much like jelly or fabric. And here you can see Joe Davis actually taking the first bite and chewing. We were really happy as well. 3 out of the 6 members of the audience that couldn't really swallow it, and they spat it out back into the plate, which was great for us because we could then show it in the follow-up exhibition, the remains of this body cuisine, which is the video installation of video documentation of the whole project, plus the bits that were spat out by the audience members. We then moved to the second in a project of those pseudo-utilitarian objects, and this is our victimless leather project. So that's in 2004. We were invited to this fashion and textile show when we decided to grow leather. We actually did quite a lot of experiments, and we realized we can grow something that looks a little bit like leather, and we were able to tan it and fix it. But for the exhibition itself, we were much more interested in this idea of showing living, or as we refer to it, semi-living leather garment that was protected by this techno-scientific body. So this idea is that when you decontextualize life, when you fragment life, life needs artificial support, but also artificial protection. So here we have something which is leather that is supposed to be put as a protection, actually being protected by our technological apparatus that also kept it alive for a while. When we showed it at the Design and Elastic Mind show in 2008 at MoMA in New York, something really curious happened. For this, we used for the very first time mouse embryonic stem cells, and they were amazing. They grew so fast that they sheared off the polymer structure and started to travel across the system, and started to clog this life support system. So in a sense, life grew out of control, and in some stage, about 5 weeks into the show, we get a phone call from Paola Antonelli, the curator in MoMA, saying something is really, really wrong. It doesn't really work very well, and she had to switch it off. We basically said, okay, you need to switch off that life support, and in a sense, killing those semi-living jacket. And you can see this is what Paola was talking about. So something that was put in a context of this very optimistic design show where the whole idea was that when you put designers and artists together with scientists and technologists, we'll be able to solve all of the problems in the world. The only living exhibit out of about 250 works that were in the show had to be turned off and killed halfway through the show. And the whole discourse changed from this very optimistic discourse into something that we like much more, and this is the contestable. The idea, we had to start to figure out what the hell we're doing with life if we need to switch it off. And it really generated quite a lot of very interesting media. I think it's the first time that MoMA was accused of killing art, and there was a murder in MoMA. And we really like this project. The interesting thing about this victimless letter that each time we show this work, something else goes wrong, but in a good way for us as artists, good way in order to generate this notion of the contestable. So this is from the Medicine and Art show in the Mori Art Museum in Tokyo. And here you can see a very young jacket, or actually at the time it was a kimono, with a small speck. So this was a really fresh jacket, just a day into putting it into the show, and I get a phone call. There's some strange speck there, which was obviously a fungal contamination. So for the first 10 days of the show, I came every day to the museum, appropriately in the show, appropriately named Medicine and Art. I administered medicine to my art, but to no avail. And this is how we then ended up as, and to a large extent, a much more interesting piece from our perspective than if we were really successful in growing the leather-like material in the shape of this jacket. It's your turn.
So I guess after so many failures, with the next project I'm going to talk about is called futile labor. And here you can see actually muscle cells moving. So when you think about, we always contesting the idea between living, non-living, machine technology versus, life, which is technologized. And one of the things that we associate so much with vitality and with our life is movement. And we decided to actually try and animate those sculptures, those semi-living, to actually, again, to make you feel uncomfortable about this division between what life is and what technology is. And of course, from an engineering perspective, we said we are going to create a muscle actuator, or we're going to try and create a little genes for the muscles so we can actually exercise them and make the in vitro meat a bit more edible, because it will be exercised like in a real body. So what you need to do with those skeletal muscles that are moving is actually to align them and try to make them work together in order to have some kind of really minimal, small actuator. And that's what we did, and this is one of the protocols that we were working with, and I won't dwell into it because we don't have time. And then, of course, you have to create the artificial body to which those things will be alive, because when you take them out of the artificial system, they die. And in the exhibition, we actually connected the movement of those muscles, which are so minute, so small that you can hardly see it with the naked eye, into actuators on the floor, so you can actually sense those movements within your body. And this a project called Fetal Labor was talking about, again, how we now have a new kind of labor, which is biological labor we know for centuries. We had slaves, we still have slaves today. But what about life out of context that is being harnessed and become completely abstract and becomes the tool and the technology of our future? How do we feel about that? And how do we feel when it will start to move and it will start to become a bit more intelligent in our eyes as we see it, and how will we feel about that? And this is again how you can see it from close up, and how those audience fitting into it. I can tell you that also we suffered from a major contamination soon after. So it was again an amazing future labor. You know,
yeah. Okay, so we have so many other projects. There's so much more context that we wanted to put into this talk, but obviously we don't really have the time for it. So let me plug. On the 26th of the month, we're going to have a big lecture as part of the RCA kind of open lecture program. So keep your ears open if you're interested in what we have to say. If not, don't bother. But yeah, 26th of January, we'll have a much longer lecture where we will contextualize our work much better. So I won't talk about this project. No, not about this project. No. But I'll talk about this, and I'll just leave you with this thought. So in 2013, I was approached by Wired UK to give them like a paragraph of what the future of materials is going to be like. And I want to leave you with this quote. As biology turns into an engineering pursuit, life is becoming a raw material. In the next decade, we will see attempts to use living and lifelike systems as smart materials that are self-assemble, self-healing, and perform different functions. Some will escape our control and will transmute into weeds and pests. Welcome to the new ecology. Thank you.
Hello. Okay. Hello again. Where was I? Okay, so I've got two laptops running here. You're going to miss out on a lot of the video, but you can go to our website at loop.ph to see the fantastic videos with sound in your own time. So basically what I wanted to talk about is the scale that we're working in our studio. We've been looking at ways of mimicking biological systems and collaborating with scientists. I'm going to keep this talk really short now because I realise it's getting quite late, but I guess what I want to talk about is collaboration and how to collaborate and how to work in this sort of transdisciplinary field and how to experiment and work outside of the laboratory, the gallery and the museum, which is what we're doing. So we're really dealing with public space and looking at placemaking and looking at the role that designers can take as being urban interventionists and working in an urban landscape. So I'm going to give you this perspective of a designer right now, and I've been involved a lot with design and education, looking how you can bring biologists into a design environment and what are the skills that designers need to be learning, because I think designers and artists are very important people. In fact, that they shape our environment. shape everything around us and the feel of things, the look of things, and I think education is where innovation really needs to happen, and it is happening in courses that have been teaching on it, so to have a play on material futures. So I just want to talk a little bit about what we're doing in the city and how we're looking at ideas around scarcity. And each year we've been working with different themes to do with food, energy, water, and more recently sleep, because I'm exhausted. And just looking at how we can bring about ideas of abundance and collaborative space. So our lab, it's a spatial laboratory. We work on an architectural scale, blowing up kind of molecular structures and building what we call scaffolds for life. And it's really our ambition to establish a completely different design practice where very different practitioners can come together in a very playful and experimental environment. And we're very much in this field of futurizing and developing different ideas about how we could be living, alternative ideas for the future. And for the past 10 years, we've been working in this context of what you would call living environments, and really looking at how we can craft space into these living, lively spaces using microorganisms, using a lot of plants, and botany's been a big part of what we've been doing, residences at Kew Gardens, and really looking at how we can harness this and looking at how it interfaces our human man-made materials. So I get the privilege to work with just some incredible people. We work with neuroscientists, actors, musicians, gardeners, mathematicians, a huge range of people. And I think maybe there's a few people here, I'm not sure, perhaps there's but really we're looking at how these disciplines interface and how we can collaborate. So we're really looking at how we can create environments and experiences using science and technology in a really enlightening way. So really we're questioning the built environment and looking at how architecture could be built. And we're really doing what we call place-based research and experimentation. And I think the design field has seen a lot of work in the field of speculative design, which is of course really interesting and something we were involved in a few years ago. But I think more recently we're trying to place ourselves quite firmly in this field of activation, of design activism, and taking these experiments into the public realm and letting them grow and leaving them there for many years and seeing what happens and creating environments where the public can come and experiment. This was an experiment that we did in the deep dark Russia. It was a place-naming project to look at how we could create this living space. We planted these lettuce on top of the building and allowed the plants that live there to grow into it to create this living scaffold. As communities, we renamed this area Happiness. It's Scheist. And so a lot of the work we're doing is really looking at the city as this metabolic system and really seeing it as a living organism and looking at how we can intervene within it. And I'm really interested in how we can transform these public spaces and really carve out spaces for dreaming and cultivating an imagination for how things could be. This was a piece that was recently developed for Canning Town, and essentially it's almost this kind of workshop space, I guess, where people come together and dream of different ideas for the future. It's called Osmo, and it's basically a huge Faraday cage, so all the signals are cut off when you enter the space. There's 9,000 stars etched onto that where you can see the night sky. Located in Canning Town. Very beautiful video here that you're not seeing. It's not so important. So I guess what we're doing is developing these tools for collaboration and using design, design thinking to bring together really divergent fields, ask questions, probe into the future. One of the very playful things we did quite recently was to work with Lucy McRae and have a lot of fun looking at what happens when two different fields come together, which is DIY, DIY fabrication and biomanufacturing. And we made a really amazing music video, which you're not going to see. You see it on the website. And the reason I show this is that a lot of our work is very serious, and we have to learn to kind of step back and have fun and play with absurdity and value absurdity. So I'm going to talk a little bit about a huge body of our work, which is looking at, originally it was looking at how to mimic biological systems, and more recently, more looking at ecologies and connections. So in the studio, we have this, we look at values from an ecology, from being adaptive, abundant, diverse, successive, and we look at how we can take these concepts into a design realm. So I was originally studying at the RCO. Oh, what's the video? Anyway, this is an animated textile. This is from like 2001, and right then, the vision was to create textiles and materials which were actually living and functioned like photosynthesis. And this thread has stayed there throughout all of the work that I've been doing since then. So this was using printed electronics, so it had a printed photovoltaics on one side and printed light emitting on the other, and it was in a constant kind of dialogue with the space. I'm kind of showing you this because we've been on this trajectory and really since that point. And it's not just mimicking discrete functions within biology, but looking much more at interconnected systems and patterns. So we have this this technique that we've been developing in the studio that's called archulates. And essentially we're looking at how to create incredibly lightweight adaptive architecture based on understanding molecular biology and carbon structures. And this is a technique we've been working on for about 10 years now. And one of the things that we really value in the studios is lightness and adaptability. So I really like showing this image where it shows how the periodic table of elements has been rearranged by Niels Bohr. And you can see he's put the lightest elements at the very top, going down to the heaviest. And then I think as designers, artists start to understand the world on a molecular scale and lightness, start valuing lightness. You can see that our man-made world right now just mixes all of these components together, whereas if we look at how nature and biology builds, it's using the top two lightest subsets. So structure has become like a real language in the studio to communicate with other practitioners, other scientists. And we're trying to understand carbon nanostructures and their bonds, and how we can create architecture. So video. And we're looking how we can create these responsive systems. So we have, you're not seeing this thing before. We have a lot of sensors on site. So this piece is constantly animated in response to the changing CO2 levels of the environment. It's a lovely video. And actually what we're working on right now is how to grow some of these structures in software, and this was a piece that was built in Amsterdam. And we're looking at a kind of a branching structure. And we're working with environmental sensors and then looking at how this data can go back into the software so that we're able to actually grow very site-specific structures that respond to the building, taking in UV, So I guess this work, again, this is a dynamic video piece, but a lot of this work really started with a project at Social St. Martin's where I was a research fellow. This is a really interesting project that looked to bring together designers and scientists. It was called Nobel Textiles, started by Carol Clay. And each textile designer was paired with a Nobel laureate. And we were really looking at the work of Sir John Walker, who basically discovered the rotary mechanism of ATP synthase, which most people here know was the first biological machine that was really discovered that spins thousands of times a second, and it's responsible for everything in our bodies from photosynthesis to respiration. And it's common in all living cells, not just humans. So it's really a fundamental process, and I think it's really vital that everybody understands this. So what we're really trying to do is bridge some of these gaps of understanding and find techniques for others to have that same feeling of wonder that we had when we were collaborating with John on this. So one of the things we developed was an experiment, or a public experiment that was called Soda Jam. And we were looking at energy and the metabolism, and looking at the whole language of food in the kitchen. So we were basically making dye-sensitized soda cells. So we're extracting dye from berries. We're making jam that you can consume and eat, and at that same time, we're using that dye to dye these dye-sensitized cells, as well as dyeing textiles. So my background is in textile design, and the dyes have been taken from berries and organic matter for hundreds of years. So it's really interesting to bring together textile designers, physicists, chemists, chefs into the same environment to explore this language. Dye-sensitized textiles. Okay, and then where are we? So, real interest in metabolism then developed into a project or an initiative that we're still working on today that's called Metabolicity. Basically, at this time, I was really researching and looking at different techniques for growing food in really controlled environments. So I was looking at a technique called aeroponics, which is a way to grow food. That's been developed in space. So it's a highly controlled environment where the roots of the plant get misted with nutrients. And I was really looking at how we could then take that technique into spaces in London and housing estates that are just completely tarmacked and concreted with very green space, and it's highly polluted, and into community centres and different restaurants within East London. So it's really looking at these kind of high-tech techniques and then how they can be brought into spaces which are kind of quite deprived and then, would benefit from some of these technologies. So this was a 2-year project that was funded by Audi. We're making cars and causing a lot of pollution anyhow. We ran a whole number of workshops where people were able to kind of really share their experiences and talk about what failed, what worked, using some of these technologies. That then went on to another initiative that was called Energy Futures, and this was in Lille, commissioned by EDF, the energy company. And they originally wanted us to create an installation that visually communicated the power consumption of the city, but we found that a little bit too sort of field-laden and wanted to really bring some cutting-edge science, stuff that's happening in laboratories that nobody knows anything about, and bring it into this public space so that people can have a much more positive vision or idea about the future. So we basically turned the park into what we call this energy orchard, and we looked at how we could augment the trees. We were looking at this new type of bark that would bioluminesce, and we just used our scenography skills to mimic some of these ideas to transform this park. But then ran a whole workshop series for over 500 children in Lille that come into this space and actually get their hands dirty or wet, so to speak, and experiment with some of these ideas. And one of them was looking at the whole development of biofacades and how you could use algae. So we actually made it a very kind of friendly and approachable technology. So people would come and we knitted these macrame textiles that were hanging in the windows out of these tubes, and we had algae that was constantly pumping through them and actually photosynthesizing. So unlike the earlier work where it was kind of electronically mimicking it, we're actually using algae through these tubes. And I'm just going to finish on this last project that we're working on at the moment, which is called the Horticultural Spa, which responds to kind of water scarcity in the city. This is kind of one of the most important projects that we've worked on quite recently. And it's kind of looking at some of the ideas that Dr. Buller had about water scarcity and how water in the future would be so scarce would be showering in these fog-like environments. So it was really looking how we could create a space, this wet space, or a public bathhouse that people could enter. And we're looking at ways of, essentially it's looking at aeroponics for people with different types of nutrients or essential oils into this fog. We had people that would come. So it's kind of a part greenhouse and part fog spa. It's on the Thames. And it was an incredibly serene space, serene, wet space. And we're now, what's interesting about this is we took an agricultural technology, which is essentially it's a nebulizer that's used for keeping plants fresh and misting plants, and then taking it into this new field, taking it outside. And we're now sort of looking at healthcare. And what's interesting is that you can actually start to impregnate fog with sort of medicinal properties, so that's something that we're also working on. It has a lovely video with sound. Okay, I'm going to stop there because we've had lots to listen to. Okay, thank you.
Thank you, Rachel. It was very, very fun. And now I would like to kind of call all our speakers for a little discussion, a little Q&A. I'm sure people have some questions. And it would be nice to discuss the collaboration between art and science and design. And also, after that, we'll discuss the future events and some of the projects that we're working on. Okay, so I'm sure everyone has a lot of questions that they can ask the speakers, but I'll just start off. I'm one of design and part-time biohacker. So I have a question regarding the relationship and collaboration of science and design. And I was asking from your experience, at what point of research or scientific project do you think collaboration of science and design should happen? Is this the ideation stage, or the prototyping stage, or the point of experiment? Refer to any example.
I think this will be the first official formula for successful collaboration. I don't think we can provide you one. What I can say is, we are very fortunate in the sense that our lab is positioned within the scientific context. So our colleagues are scientists. The more years we working together with them, it's become we collaborate even about discussions about the philosophical and ethical implication of what we work. Again, I think the problem starts when artists think that they are doing science, and science thinks that the scientists think that they can do art, and then usually, that ends up not so good art or not so good science. But again, I think all people are, if there is an interest in the same issues, and also we are again fortunate enough to have a lab that is dedicated to artists and people from the humanities to come and actually learn the techniques themselves. So then not only they can do some of the work themselves, they can then talk to their fellow scientists with a bit more knowledge and the jargon that you need in order to feel secure about that.
Right. It's kind of interesting because obviously Symbiotica is already a fixture within the university, within this biological science department. We don't really feel that we need to justify what we're doing as many other centers and places do. We see ourselves as researchers that are embedded within this context of figuring out what's going on with life. I must admit, as the director of Symbiotica, I abandoned the term art and science collaboration quite early in the game, realizing that there are issues around the notion of collaboration. Now, also something to remember is that the term collaborator wasn't a very positive term, let's say, in the Second World War. Yeah? So you have to think about it in those terms as well, that, you know, it might not be the case. It is about when we work with scientists, usually when you get artists that have no scientific experience, we get the scientists to mentor them. They're not collaborating with them, they're mentoring them, so the artists can develop the skills they need in order to pursue with the projects. Because as I mentioned, we're a staunch believer in this experiential engagement. The artists have not just think about the idea, they have to deal and work with the material, especially when it comes to working with living biological material. There's another layer of engagement and responsibility that one needs to exercise when you work with living biological systems. Now, when it comes to what you refer to the stage of the idea or whatever, you know, in most cases, actually, in our cases, the lab is set up in such a way that it's an artistic research lab, and therefore the artists are privileged, and the artistic research is taking the precedent. There are secondary outcomes. You know, for example, we never tried to be the first people in the world to grow meat or leather. We've done it because we're interested in some of the questions that leather and meat grown in such a way generates. So whatever other people would pick up of that, those are the secondary outcomes that, again, we are fortunate enough, we don't really need to celebrate those things because we're already so ingrained and embedded within this context. Now, I know that with biohacking communities, with the initiatives that are just starting up, there's a need to somehow justify and promise those types of outcomes. But in most cases, what we've seen, the aesthetics of disappointment takes over, and those expectations are not being fulfilled.
Putting on a bit of an industrial hat here, responsible innovation has become a bit of a buzzword in the field of synthetic biology. But at its heart, it's simply about looking at the broader context of what your actions do. And to be frank, that's just good business. You know, if you're bringing a product to market and you don't understand who it impacts, well, you are going to be blindsided by the risks that you're walking into. And art can often be a very effective way of understanding through imagination what are those risks, what are the ways that people are going to viscerally react. Because the problem with biology is it's inherently dual use. Nature is not always friendly to the world. And understanding those implications, positive and negative, so that you can address them, so that you can make sure that you are balancing your actions towards the positive effects, is one of the things that working with art can help provide.
When we talk about positive effects to whom? And just
I agree, broader context.
I mean, I guess we've always been really clear that we're working in terms of like public engagement, and we work, you know, with the scientists and the institutes quite often our client, and we're finding ways to communicate their work. And that's quite a traditional role, I think, with art and design. And it's kind of a really exciting time right now where there's a real fusion. There's this one interesting story where we worked with the mitochondrial unit in Cambridge to try and explain some of these very complex ideas, and we had a particular technique which was based on making, a textile making technique, which was just really embodied, and it took this group of scientists away from the computer and their just traditional ways of working, and we were invited back to this group of scientists, an incredible group of people, and ran a handcrafting making workshop so they could understand molecular structures. And then that was the first time for me that I felt like I was actually having any interesting role or interesting exchange, and it was completely on a kind of geometrical and structural level. So I guess there's very different ways that you can play a role and work together.
All right, thank you guys. First, I just want to say everyone's talks have been really interesting. Everyone's kind of approaching biology from like a very different angle. So I guess what I'm interested in is that it seems like, you know, biotechnology over the last 10, 15 years, you know, the cost has come down. It's been more accessible than ever. You see artists, you see designers coming into the field. And I guess the question is like a question of like bioethics, like when do we, or like how do we regulate this kind of activity? Because I can imagine, you know, once like the language is there, once synthetic DNA is so easily producible and so cheap that, you know, anyone can get their hands on it, and anyone can create, you know, whatever they want to create. So like when it comes to regulation, what do you guys feel?
Well, I was actually at the UN last month for the Bioweapons Convention, because those are the exact questions that are being asked by regulators globally right now. But there's one very simple point. Can we regulate disease out of the world? Biology is inherently a dual-use technology. And what we need to try to do is embrace the ability to work with it so that we can respond resiliently when things happen. Because it's not just a case of wondering about what does a rogue person in a biohack space does. It's about what's going to be evolving in the Congo next. It is about where does MERS goes pandemic. It's about when does Zika or the next mosquito-borne pathogen cause wide-scale distress to populations and death. So it's not just a case of saying we can put this genie into the bottle, because life is all around us. This genie is there. It's about understanding how we can rapidly respond to what's out there. And that's the only true security that we can possibly have.
I think it's a really important question. From my perspective, it's more to do with mindsets than the actualities, because to a large extent, with all due respect to all of those advances that are happening, I think that we are really still in the stone age of biology. We're still, in a sense. But basically, so what we see now is more what mindset is driving us towards those changes in our relationship to life. Now, if there's anything that we can learn from history, is that what we choose to do to life, we end up doing to ourselves. You know, one of the things, and this is something we'll talk about on the 26th, this whole notion of cultural amnesia that we went through, that forgetting that actually quite a lot of the discourse that's happening now happened in the early 20th century with the same type of assumption that we are going to have control over living systems in the very near future. Now, regardless of that, I think what we need to do is we have to be not just resilient in regard to the type of technology we have, but also in the type of mindset and the humility that we need to exercise when we're starting to engage with complex living biological systems, that we have no idea what the cascading effect of those actions are going to be like. In our perspective, it's really interesting because obviously there's an assumption that artists are much less responsible than scientists. So our work is being scrutinized much, much more than our scientific colleagues within the context of the laboratory. So we need to constantly prove that what we're doing actually fulfills all of the regulatory requirements and all of the requirements of the institutional ethics committees and so forth. So we adopted the language that also changed the mindset of the committee and the institution at large in regard to what can be done and what we think about life in this context. Now, I'm really concerned about some issues to do with over-regulating, but at the very same time, I think that the precautionary principle should prevail over the reactionary principle that seems to drive quite a lot of what we see coming out of the west coast of the United States, for example. I think we still have to maintain some form of a reactionary principle in the context of biology.
Well, I take pleasure in filling in the ethics application form. I think many people know this. I very much enjoy that challenge. And what I find is that because of the way that as artists our ideas develop, that quite often we're asking if we can do things that they haven't even thought of anyone trying to do. And so the ethics aren't out, they haven't caught up. So I find myself sometimes having to self-regulate because the regulation doesn't exist, which is quite interesting. I'm talking about particularly in the display of plasmids. that I'm working with highly pathogenic. Well, they're not pathogenic, they're just DNA, legally. Plasmids, so things like that. So I find that what I'm having to do is kind of raise issues that the scientists I'm working with feel uncomfortable doing, but aren't anything problematic in terms of regulatory stuff. They're allowed. So I could just do it. But do I want to do it? No, I probably don't. I want to make sure before I do it. But so I'm stopping myself, like yeah, but I don't have to. So that's when it gets into an interesting kind of ground that we could be doing it. And then you've got, you know, the CRISPR-Cas9 sort of stuff. And I was talking to some leading UK scientists in public health, and they said, well, you know, they're looking at it in terms of how you regulate that. But of course, they said, of course, you can't, because you could just do it in your shed. So if you have the expertise, which, you know, doesn't take that long, but it's not something that artists going to art school are going to come across and just suddenly stumble across either. It's a little bit in depth. So I think that focusing on artists as a particular issue, or biohackers as a particular issue, it's part of a broader question. And basically, ethics is the thing that happens in the moment where we don't know the answer. And that's why it's interesting. But it's all in question as well for grabs, really, in a kind of horrible and terrifying way.
Yeah, so I guess to kind of go back to the question, engineering biology, it's indisputably the most powerful technology that any of us will ever use. Does it need to be regulated? Do we need to be careful about how it's used and what we use it for, of course? But just to reiterate, I guess, Sean's point there, viewing this in the context of the world that we live in, where we all know people who have been affected by disease. We all know that there are illnesses and diseases that are currently ravaging the world. So it's equally important to take into account the fear of the unknown unknowns against our ability to kind of turn our minds off to the things that are going on right now, which this technology can address.
By the way, I've got a book on this subject. It's called Trust Me, I'm an Artist. You can find it by my website.
Yeah, it's interesting hearing everyone talk about this issue of bioethics and kind of you know, everyone's like hearing like these different mindsets and different I guess like philosophies on where they think biology will go and what they think biology should be. And I think it's really relevant now that, you know, like BioChanges, you know, biology is becoming this global community. It's not just biologists, it's not even just engineers and scientists, it's, you know, a whole bunch of people. And so I guess like kind of moving forward, what do you guys see are like the biggest obstacles that like this bio community faces, that I guess like these kind of new technologies that are emerging faces?
Working with biology costs too much, takes too long, and fails too often.
Almost took the words out of my mouth. That's interesting. Yeah, because I think one of the things is that life fights back. Yeah. And as I mentioned, this idea of the new ecology where whatever attempts of us to control those systems would undeniably fail in some way or another, especially when you start to release those things into the environment. By definition, life is ever changing. Yeah, so if we take that in mind, if we try to be more, in a sense, collaborative with life rather than try to impose a will on living systems, we might see ourselves going into interesting places. Now, United myself wrote a polemic against engineers, and sorry for the engineers in the room here, but this idea of the engineering mindset taking over control over the direction in which biology and biological technologies are taking might not be the best way to continue. And obviously there's other models that we need to explore in regard to how we engage with other living systems. So another thing that I suppose we need to talk about is that trying to cluster all of those different groups together, which is great to see here. You know, it's a great way to communicate. But I'm a staunch believer, and especially in the last few years, especially since the growth of the biohacking community, that artists should be treated somewhat differently because artists are professionals in their own field. We're not hackers, we're not amateurs. What Jonathan and myself are doing is going around the world and setting up professional labs for artists to engage in a professional way with biology rather than this hacker type of amateurish, not that I have anything against it, mentality. Yeah, no, but basically what I'm trying to say, we're not amateurs and don't treat us as such. We are professionals, artists or designers working with the new technologies in a professional way, which don't have to be clustered necessarily with the whole kind of biohacking mentality. Very, very different mindsets in this context.
I can just add about the difference because it's not about amateurish versus professional. As much as, as artists, we are interested in the cultural aspects of this technology. It's not just about, forgive me again for being genderized, like boys with toys. That's not what we're interested. We are much more interested in, again, the ontological ideas, the ethical, the philosophical. How do we engage with the audience rather than sitting and trying to build things just for, maybe one day we'll create the next, I don't know, IP or whatever. So this is, I think, another difference that we would like to emphasize.
It's contestable.
It's contestable. And again, yeah, let's put it that, you can answer back, please, by all means. We want you to actually confess that. Here we go.
One of the questions I had was sort of related to this point. So you're talking about how it's not just about what you make, but how people react to it. So for example, with the in vitro meat idea, for example, I mean, over there, it didn't look like it was made to look tasty at all. But was that like a conscious decision? I mean, did you want it to look like real meat? Or maybe the question that we should be asking is, should fake meat try to even look like real meat? And so how do you draw the balance between what the science is doing and how that helps society, and how society can adopt this kind of new technology in the real world?
How long do we have? The story of in vitro meat is a really interesting story from our perspective, and there's a really kind of large area of interest. But we don't really see ourselves as doing public engagement for science communication. So our role was never, and in the context of the in vitro meat, was in particular, it wasn't about doing something that looks tasty. It was, as I mentioned before, engaging with this idea of the relationship, this intimate relationship that one forms with life once you consume it. And it's never, there's embedded violence in that, regardless of what you do, that there's violence exists within us consuming another living form or another living being for our survival. We have to acknowledge that. So deliberately, we didn't really want to make it beautiful. That wasn't really our interest. And actually, as you've seen, most of our work, the aesthetics are not really about, or how shall I put it, we use other strategies of seduction rather than beauty in our work. And we deliberately so. Yeah, there's other kind of aesthetics to it, but that's not the case. But the interesting thing with the in vitro meat project, and again, I think on the 26th we will talk more about it, is that at the time it was a one-off project that we didn't really think that we were going to continue. But as in the last few years the story continued, we're now developing series of projects that are responding to what happened, 15 years hence, and how the narrative, because the narrative of in vitro meat at the moment is extremely seductive and problematic, because from our perspective, it's not a solution to any problem. It's actually a symptom of some of the issues that we're facing. And I suppose I'll talk about it more the other day, because I just don't want to get into it too long now.
Having eaten some of the current generation of in vitro meat actually is quite tasty.
Which one you had? Okay, I had the modern meadow as well, the second generation. It's tasty because there's artificial flavors there. Yeah. Rather than the meat itself, so we can make a piece of sheep taste as well, if you put the right flavors in it. That's not really what's going to solve the problem.
It's cultural, it's relative, If someone wants to sell you something, they'll do anything that you'll find, that you'll desire it. We're very malleable. You don't know, what you eat now that what it actually consists. So I think when we talk about taste, it's very relative.
Do we have any questions from the audience? Okay. I guess, is there one? Yes? Okay.
Hello. I'll just related to the last point, I think. How do you think engineers can get involved or be useful and not get their engineering mindset get in the way?
You know, I like engineers. They make great servants. Boom. No, but the reason for that, okay, here we are. Sorry. No, the engineering mindset is extremely important, but the problem with engineers as a mindset, not with individual engineers, you know, the humans like any other humans, is that the profession of engineering is about trying to find the optimal solution to a set of problems. And in most cases, with kind of trying to avoid any other noise in the system. Yeah, anything that disturbs this optimal solution is being kind of discarded in order to achieve that. You know, and you need to do it when you build a plane. You don't want to have the noise in the system. You want the plane to fly and not crash. And when you build a bridge, you do a similar thing. When you start to work with living biological systems, when you start to work in complex systems, when you start to serve agendas which are beyond just optimizing a system for a specific outcome, it's becoming too complex for engineers to deal with, just by the nature of their profession. So I think engineers have to always work in teams of people who think about issues in a different way, in a more kind of holistic way, if you like. People like us, what we're trying to do with this idea of the contestable design, to work with people who constantly question the engineering mindset, constantly question, okay, you found a solution for this problem, but what about the other problems that this solution created? How are you going to approach them? How are you going to deal with them? So it's always have to be in concert. And my concern is that, especially when it comes to the discourse around contemporary biology, is that the engineers saying, no, we are going to call the shots. We are going to run the show. And this is the issue that I feel that we need to figure out ways. You know, I'm doing it in a contestable way. I'm sure that friendlier people would do it in much more in other ways that would actually want to antagonize engineers. But what I find actually that my contestable approach seems to trigger engineers into working with me because I challenge them. And those types of challenges are extremely important for all sides. So they're playing a major role. I think one of the issues is that many of the engineers that are currently working with biology, especially the first generation synthetic biologists, who came from things like mechanical engineering and electrical engineering, have very little idea of the actual making or what really makes biology tick. And they're trying to force their assumption of systems over biological phenomena that doesn't really fit very well. I would claim the second generation, the third generation, people who are finishing PhDs now are much more nuanced in their approach to life, and I think we'll see much more interesting stuff coming out of that. But in the big debate between kind of the engineers and the biologists, at the moment, those who are calling the shots are people who have never been trained as biologists, and I think they have to be much more humble. And I think they're becoming, whereas they realize that the biology doesn't operate, as you said, doesn't really operate in the way that they assumed it will, because they just read the short synopsis of what the biologists are doing.
I absolutely agree. The biggest mistake is to apply mechanical engineering to biology. You know, it's living systems. It wiggles, it jiggles. It's probability functions. It's that complexity that gives it some resilience. Yet, for all of the things going on in the molecular level, when you tell your arm to move, it normally does, unless something else has interfered with it heavily as a system. Biology still has structure in how it operates. It's just that you need to unpick the complexity that helps drive that structure. And it's beyond, to be frank, the imagination of what we can look at in any sort of isolated context. The way we're being successful in engineering biology is, in fact, to embrace far more of the complexity that makes a system operate. It's, in fact, how do we look at more of the context of a system? Because it's only by looking at the full context that we're able to start to determine how does this act reliably. Because you see examples of reliable biology all around you. You have 10 fingers, you have 10 toes. You're able to generally speak and communicate. You see systems having this emergent complexity all around you that ends up having reliable function.
Yeah, so, but the question isn't, the question I put to Joe Andy as well is, are you just, if you still maintain some kind of the engineering mindset, are you just making the black box bigger?
I would say you're definitely acknowledging there's a whole lot more knobs in it.
Do we have time for another question? We have questions from the back. Yeah.
Hi. We heard a little bit about what you saw as the main challenges ahead. I'm just interested to hear about what you see as the main risks ahead, considering the expansion of that field.
They come from many places. One of the first ones is, of course, that we're developing technologies that allow unilateral action now. One individual can, using a technology like a gene drive, create an impact on an entire ecosystem. And it's almost an inevitability, given that potential power, someone will use it. But I repeat the prior statement, it's like asking, how do we regulate disease out of the world? And I'm incredibly concerned that our fears about what can be will also prevent us from following the hopes of what can be. That we lose in our concerns of risks the also important narrative about the benefits.
I must admit that, sorry. Yeah. From my perspective, it's basically the business model that drives contemporary biology, which might be the greatest risk ever. So at a time where we're starting to figure out ways to control biological systems, or even not control, but claim ownership over biological systems, we're also at a time where this idea of ownership is so prevailing, and it's kept in the hands of the few. So just to give you an example, there's companies now that are trying to re-engineer our microbiome. And we heard about the fecal transplant. What about, and there's other companies trying to figure out ways in which they can read very, very fast the outcome. Yeah, so you would imagine that in not so far future, we'll have toilets that would read your feces and will tell you exactly what microbiome you have. At the very same time, they'll also read those feces to see if you use proprietary bacteria. So you would have companies that would force you to pay royalties for your shit. Yeah? Because if you continue with this type of business model that drives our relationship to biological systems, that's what's going to happen. And this is a very benign notion. But more than that, it's this idea that maybe the marriage of our current neoliberal mindset and our increasing power over the biological system are not going to be compatible for most of us.
Following up on the microbiome stuff, yeah, that won't work. Absolutely. I mean, the biggest risk is companies like BioMeetup doing this like advertising that they can tell you if you've got the microbiome of a runner or an alcoholic or something like that. It's literally guesswork. They can't tell you anything. It's really minimal, the information. They've no idea of all the complex interactions going on. With the human fecal transplants, basically the science is this: they know it works, but they don't know how. They literally do not know how. It's authorised for use in the UK. It has been since 2014 in extreme cases of patients with drug-resistant Clostridium difficile infections that have failed to respond to antibiotic treatments over a sustained period. Literally, these people have no other option. And at that point, it's authorised for use in the UK. And the idea that we can tell what's going on in these complex ecosystems, even if you sequence the entire genome of every bacterium in your body, I still think we wouldn't know what was going on really. It would be such a complex thing. So part of the problem, I think, is the bullshit and the naivety of people. And I think the challenge, partly on the public engagement front, and we've debated the issue of public engagement before, and I think that my work is partly in that, because I think it's a political issue as well. It's that companies are trying to sell us these ideas, and the public don't have a way of deciding what's rubbish and what's good, because you've only got the things like the tabloid newspapers to go on, and that's the biggest risk. I think it's the manipulation. I think it follows on from what you're saying, the manipulation of the public, and people trying to sell us things because we're naive. It might as well be snake oil.
Should we wrap up now?
All right, so I'd just like to thank this really great speakers again. Thank you guys for coming and sharing with us what you guys do. And hopefully you guys can stay in touch with BioChanges as we move forward. Just before we say goodbye, I'd just like to make a few announcements. We have a couple future events. The next BioChanges event is going to be in March, based at UCL. And right before that, we're going to have a talk by William Myers, who's the author of Biodesign, coming on February 3rd at 10:30 at the RCA Lecture Theater 1. So you guys are all invited to come and join us there. And I believe the collab, yeah, if you'd like to make it now.
Hi, so you see in the bottom, that's our logo for now for this workshop called Collab. And Paloma here and I are part of this association called Open Science School. And as we are using Open Science School to create this event called Collab, where we invite literally anyone interested in biomaterial. And we have it's a 3-day workshop. And by the end of the third day, we have managed to book Institute of Making, which is a workshop in UCL. So then through these 3 days, we really want people to think about not just about the future of biomaterial, but to play around with biomaterial to then have interesting discussions going on. So if you're interested, I have flyers, so come find me. Thank you. And the last thing, also at the MIT Media Lab, there will be one of the BioChanges talks, and it will be hosted by Joe Davis, who's, I believe, a good friend of some of the speakers. And yeah, so if anyone's around in Boston, feel free to come. And also, hopefully, it will be a live stream so that we can look at it as well. And also, because it's a community, there's some of the projects that the students are working on. So if there's anyone who wants to collaborate or work together with us, please get in touch. And that's why we have little slips for you to leave information so that we can start sharing information and meeting up and working together. But in general, no, thank you so much. It's been great. And it's such an amazing thing that we're growing and there's more people and more interest. And we have such a great speaker. So thank you very much.
I don't know how to do it though.
Oh, have to transfer your stuff.
Yeah. And this, we transfer it, but it's taking
It's okay. You have the
Pronto?
Well, I work around the corner, so
Yeah, actually. The thing is that I'm over at UCL. So I could contact these guys in the area and they can do that. Are you in a rush?
902.
Paloma? Yeah, I'll send you a message. And what do you think of the event?
Yeah, I think I really want to see the simple stuff, but I missed it.
Really?
Yeah, so,
Are you interested in it?
I've got big interest.
Not necessarily engineer?
Well, I studied physiology. I didn't do it. I really enjoy science. I choose science
But then not really actively working.
like maybe when I retire.
Or BioMeetup is a really good place for that. Have you been? You should go. It's just nice if you actually do have an interest, be with people that have it as well, that are in like practice or like working in like a private company. Yeah, it's really, really great. Like the atmosphere is really want to play around with it, not really serious. It's really great. It's meetings are on Wednesdays and Sundays, so tomorrow is open to like anyone. But also Sundays, tomorrow will be like 7:30 or 8, and it's quite a bit easy now, it's gone, but you can also make some just regularly go and have access to the little lounge. No, that's Hackney, it's a bit far, but oh well. Exactly, it's overgrown. But I'm thinking what to do with the videos. But then, I mean, as in how to get them from the Etsy and then give it back to you. Maybe someone here has some storage space. But if you're in a rush, don't worry. As in, yeah. Like on my side, there's no rush. It's just that it would be great to get it at some point. Nice. Yeah. Brilliant. Thanks so much. It was really like a lifesaver, just like really gone otherwise.
Guess it was meant to be.
Yeah. It's a nice idea though. I will start carrying an SD card with me everywhere.
Yeah. I make film though.
Huh?
I make film as well.
Normally.
Yeah.
Oh, nice.
I shoot film. I've got a Canon just like yours.
Really?
Yeah.
So it's more like just like independent.
Yeah.
Whatever you like.
So I just have actually I was editing last night, that's why I still got it on me.
And what are the films about generally?
Generally about music.
That's amazing. Can I see something?
Yeah. Send me a message on WhatsApp now.
Exactly. I'll say, I'm Paloma, I'm practicing videos, and then at some point, I'll come to Imperial and get the videos.
Cool, Paloma.
It's nice to meet you, Anthony.
Yes, pleasure.
And do go to my house.
Did you send me a link on WhatsApp as well?
Yeah. Brilliant.
Alright.
Really proactive interaction here.
Exactly. That's what science is about, right?
It's physiology.
Right. Cool. Take it easy.
Yeah, it was like really good. She had like bags and chicken feathers, bags of carrots. She was like, super engaged. It's good learning that person. Nice! You're letting the mud see. Oh shit. Yeah, there goes the that's sweet. No, it's okay. That's not Damn.
What did you get your nose from the camera?